For the continuation of our topic, we'll be moving on to the regulation of the cell cycle. Mistakes in duplication can lead to mutations that can lead to cells being abnormal and develop into cancer. Because of this, cell cycle checkpoints are done. At checkpoints, the progression of the cell cycle is halted until all parts are checked and conditions are correct to continue with the duplication. Checkpoints are at the end of gap 1, at the gap 2, and M transition, and during metaphase. At G1, there is a cell growth checkpoint. This check occurs at the end of the G1 growth phase. During this checkpoint, it checks whether the cell is big enough and has the proper proteins and nutrients for the synthesis phase. If the cell is not big enough or not ready to go on the next phase, they enter the G0 phase as they can see or as you can see on the diagram. So here is the G0. In G0, the cell is in the resting phase until it is ready to move on. At G2 checkpoint, there is a DNA synthesis checkpoint during the S phase. Here, there is a check to make sure that the DNA has been replicated correctly. If the DNA is replicated correctly, it moves to the M or mitosis phase. The final checkpoint is metaphase checkpoint. This occurs in the M or mitosis phase and checks whether metaphase is complete. If it is complete, the cell divides and the process repeats. There are genes and proteins involved in the checkpoint. They also regulate mitosis. The best example of a protein involved is the p53. Any segment of DNA can be damaged by heat, radiation, and chemicals. But p53 will notice this damage and stop cell division. It then triggers enzymes that will repair the damaged region. So this is the cycle of the CDK cyclin complex, and it's good to know that CDK is always within the cytoplasm. It doesn't disappear. The number of cyclin goes up during interphase, which is the longest phase in mitosis, and the amount of cyclin reaches, and after the amount of cyclin reaches a certain threshold, mitosis is triggered. CDK, or the cyclin, for the information of everyone, is like a key in turning on and off some of the enzymes that are involved, or the proteins or some gene factors, so it can trigger mitosis. The number of cyclin is a great mechanism the cell uses to regulate mitosis activity. So to begin with, some examples of genes and proteins that regulate mitosis, we have the following. So the first one is the CDK1, and CDK stands for cyclin-dependent kinase 1, a central regulator of cell cycle progression, particularly at the gap2 or metaphase transition, and its activity is dependent on binding to cyclins like the cyclin B. Another type of cyclin is the cyclin A. It plays a key role in the gap1 or synthesis transition, activating the CDKs to promote DNA replication and initiation. The third type is the cyclin B, which is essential for entry into mitosis, forming a complex with CDK1 to trigger mitotic events. The fourth one are known in the acronym of RB, which stands for retinoblastoma protein. It acts as a tumor suppressor by inhibiting E2F transcription factors, preventing uncontrolled cell proliferation during the gap1 phase. This is the most famous, should I say, or the most useful type of the genes, which is the p53. P53 was actually named named the Molecule of the Year in 1992 for its contribution to cancer research. And it is also believed that P53 is essentially the toolbox of the cell. And another term for P53 is the tumor protein number 53. It is a critical checkpoint protein that responds to DNA damage, triggering cell cycle arrest or apoptosis to prevent mutations from being passed on. It's very important to know that if the DNA is properly repaired, P53 will allow for the DNA replication and cell division to continue. However, if P53 determines the damage is beyond repair, it will trigger apoptosis or cell programmed death. Let me give you an example. When your skin peels off, it's because of apoptosis. So you're actually witnessing P53 at work. And because P53 is so important, it can cause problems when it is malfunctioning. And it will fail to stop cell division or repair DNA. Instead, the cell will continue to divide with damaged DNA. If this damage accumulates, the cell can turn cancerous. The last type of protein is the CDC6 or the cell division cycle protein. Involved in DNA replication initiation during the S phase, essential for proper DNA duplication. So each of the 6 key genes are working together so that they can regulate the division process. On the other hand, if there are mutations in these 6 genes, cancer is likely to develop. Please keep in mind that these genes don't cause cancer. When turned on and off correctly, cancer does not develop. It's when these genes stay on or off that causes the issue. And some other proteins that regulate the mitosis are the following. So when the growth hormone or the growth promoter genes is on, it can result to unlimited growth. The tumor suppressor gene, or known as the P53, if it is off, it ignores the checkpoint stop signs. The apoptosis genes, if it is off, the cell doesn't go through apoptosis. Chromosome maintenance genes, or known as the telomerase, if it is on, it will undergo or allow unlimited divisions. The touch sensor gene, if it is off, it overcome density dependence. So it's very important that the cell division is being regulated with a combination of the key 6 genes and the proteins. Cancerous cells are known as disruptions in the cell cycle. They are the cells that are unregulated and they divide uncontrollably. Cancer cells have the ability to migrate to other regions of the body and they didn't originate in. On the other hand, apoptosis is programmed cell death. It is a normal and controlled process within multicellular organisms. It is very important because it maintains the balance of cells within an organism. And apoptosis happens when the lysosome bursts, causing the cell to burst and die from the acid. So even the death of some cells contribute to homeostasis and regulating the process in the body. For example, apoptosis can occur during the development of new cells. If these cells are not divided correctly, they could lead to cancer or a virus. Without apoptosis, these harmful cells could continue dividing and multiplying, which would cause an adverse effect on the organism. Apoptosis is very important because if damaged or mutated cells do not get the signal to go through apoptosis, cancer can form, which is way worse than any result. For better understanding of the cell regulation, I provided an interactive link for you wherein you can click it and play around with some of the regulation of the cell. Should in case you have questions, please write it on the lowest portion of your notebook and let me address it when we resume with the class. You have a good day.